Hi, so I work at the CIRE, Centro de Investigaciones sobre Desertificación in Valencia, and we are studying plant communities and how species interact mostly in semi-arid Mediterranean climates and mostly shrublands. So what I want to talk about is a work we've done and that is published and will continue using generative AI. And the idea is to study plant communities and how the species interact and use that to help plan restoration of ecosystems in common mines or other areas. So I want to go quickly over some introduction about variation of autoencoders, which is the formalism we use, and then show you a little bit of how we use them to try to learn the basic rules that underlie species coexistences in the ecosystems we've been looking at. And specifically, we've been looking at a data set that some colleagues of ours have collected near Alicante, so in southeastern Spain, near Petre. And the next step was, of course, to try, there's two things we want to do, which is predict what are the best strategies for restoring ecosystems, and in particular, not just for that place in southeastern Spain, but to transfer that knowledge or these basic rules that we hope to have learned to other communities. And so I'll show examples where we try to apply this to southern Spain, so in Murcia, and another one in Mexico. Okay, so variational autoencoder is one specific type of neural network. And the typical way it is used is that you feed it an image, and you try to reproduce that image. But the aim is not to reproduce that image exactly. That wouldn't be particularly hard. You wouldn't need a neural network for that. But it's to have these different layers of neurons, and in the middle have some random effects. So you have this, in these various layers of neurons, there's one that's called latent space. It's a layer with an arbitrary number of neurons. You choose the dimension of that latent space. And they're actually, the output of these neurons is just a sampling from the previous layers to give you the mean and the variance of the distribution. So you sample from Gaussian distributions. So that's the stochastic part, if you want. So it's not completely deterministic. And that's very important for the rest. So that kind of splits your neural network in two parts. The encoder that takes the original information, here an image, and encodes it along these 2, 3, 10, 20 dimensions, as many as you want, and gives you a representation of some variables that summarize and describe this image in a different way. Instead of brightness of each pixel in each color, you can summarize it, you can compress it, for instance. And then you have a second part that decodes that information again, and presumably reproduces pretty much the same image, but never exactly the same image, because we've introduced some random variability. And the reason we introduce, or people introduce, that random variability is that when you look in that latent space that here is represented as if it had 2 dimensions, you don't want each image, an image of a triangle, of a circle, and of a square, to be just on their own, some spots, some infinitesimally small places in that space, but you want to have a continuous space where the distributions for each of these images overlap, so you can move around continuously and not have gaps where in that space, in that hidden layer, that latent space, there is actually no information stored. And then, if there was no information from your original images stored, you might find like this purple wiggle. Oh, the pointer there. So, the point of this is that then, when you move around that space, these latent variables, you hope to have a continuous variation between, let's say you have these 3 distributions, these 3 colors here, so these 3 Gaussians representing 3 input images. If you move around that space, you have a continuous variation. That's the hope. That's why you regularize this space with a pullback divergence. Okay, so that's all I want to do about. I want to say about the technicalities of the neural networks. But again, the point is to reproduce reproduce the images, but having simplified and smoothed the structure internally in terms of some new variables that we hope, or we believe, are deeper and more meaningful and have a more human interpretation. The effect is, for instance, if you do that with images of handwritten digits, so this is a famous problem, when you move around that latent space here in 2 dimensions, you can go continuously from a 1 to a 7, at the bottom, 1 to a 7, and you have this intermediate digits that never really existed, nobody ever really used. Here, there is a slanted 1, nearly a 7. And so, the point is, if you want to think about it in terms of, let's say you had an image of a dog, a bird, and a car, it's a vector with 1, 2, 3, maybe 8 dimensions here. But there might be 2 dimensions that are easily interpretable. One would tell you that the other can fly, and the other one whether it's alive. That's the hope. That doesn't always work so well. And so these variables in this hidden layer are special because they organize the information in an interesting and very useful way. So a bit more fundamental. It's been used with images of people, and these latent dimensions are then associated to having a smile on the face or being a man versus a woman. So you can shift gradually and smoothly from one to the other. Okay, what does this have to do with ecology? So we used these variational autoencoders in a paper we published last year with some colleagues in Valencia, some of them physicists, some of them biologists. And the idea we had there is to study vegetation patches. So it's, this is not the exact place what the data comes from. I didn't have pictures from that place, but it's a typical Mediterranean shrub vegetation. And you can see that the plants are clustered, clumped together in patches with, I have a more zoomed in picture. There might be a bigger one that is already adult, that is the nurse, acting as a nurse to facilitate the growth of others. And you have some smaller young recruits below. And so the question is to understand which plants facilitate the growth of the other ones. So now, when I showed before variational autoencoders, I was talking about giving them images and reproducing these. Here we're not going to feed these images, that's not the point. We're not trying to recognize the species on the images. So how do we use this formalism? We have plants and there's variational autoencoders where I typically used on images, so it's like fitting a square peg into a round hole. So what we did, and this was not my idea, it was my colleague's idea, is to encode each patch, which is a combination of plants, as a barcode. And the bar is either 0 or 1, depending on whether that species is present in that patch. And we focus on the most common species. So for instance, this barcode would tell you that the first one is Esparto. I don't know, it's maybe a typical plant from around the Mediterranean, is absent. The next one, Elianthemum ciliacum, is present in that patch. You could generalize this to not just presence absence, you could count the density, the number of them. And I'm running out of time. So you're feeding that barcode in and seeing what comes out and trying to optimize. You perform gradient descent and all this kind of thing until you minimize the cost and you get as good a result as you can. And then the system presumably learns all the distribution. These are the patches, 2,000 and a few of those. With here only put a few species, but in the end we work with 16 different species. And the model hopefully learns what are the chances that this species is associated with this one. And from that we want to predict what happens when we put one seed of a plant or a species. So here, let's say I will feed it patches that are artificial, fake patches, synthetic patches, where there's always Brachypodium reticum with 100% probability and some seeds of the other species. So just some small likelihood that the other one is there. And ask what actually comes out, what grows. So I feed it to the variational autoencoder, and out comes the Brachypodium reticum, the second from there, always there. And of the others, only a few grow, and I have the probabilities of each of them growing. So from this, we claim, or we hope, now I want to test it, that when we go and perform restoration in these areas, if we plant this or that species, we know what patches of vegetations are likely to grow and which ones are the most diverse. In that example, the species at the top show you that if you plant that first species, the one that's at the top, there's only 10 different patches that are produced. Right? Whereas the one at the bottom will have a large variety of 50 different patches possible. So that sounds like a good species to generate biodiversity. And I guess I don't have much time to go into the rest. Okay, I'll just show what we want to apply it to. Then we want to transfer that same model to another place in Murcia, so a bit further south in Spain, a few hundred kilometers south. But this is an abandoned mine, so it's actually quite different. Different species grow there. And then yet another system in Mexico. There is also semi-arid, but quite different plants, more trees as canopies, and cacti growing underneath. You might be surprised to hear that it actually works after some tweaking. We can actually learn some rules from Alicante, let's say, apply it then to Murcia, and there is some information that still works, that still transfers and gives us some result that is better than random, quite a bit better. And then we can learn on top of these basic rules some local rules by doing some fine-tuning of the models. So transfer learning, if you take the model we trained in Alicante, we apply it to Murcia. It does give us already a result that is better than random. And then we fine-tune it. So we train it further on the local data, and we get an even better result. We try that, it does work, and then we try it even on an even more different ecosystem in Mexico, and it transfers as well. So we're quite happy about that. And now we want to check on the ground if our predictions of what pioneer species will plant predicts what species will grow next. If that is verified, and if it works, then we use it to plan future restoration projects. I think I'm out.